Professor Lumsden will now make his initial 45-minute presentation.
I only thought you had to build your own classroom at Tulane. Then I went to ICR, found out we had to do the same thing there, and now we do it here at Wesleyan as well, so I feel right at home. Preliminary remarks. First of all, I want to thank Professor Hanson for putting this together. Thank you folks for showing up tonight to listen to us. And I really do appreciate being here. And I most certainly appreciate the spirit in which Professor Hanson put together this intellectual discussion. What we're really here to do, of course, is to discuss ideas. As I've been telling my students all my life, don't ever be afraid of an idea. Listen to it, examine it, sort it out for yourself. Use, if you will, your God-given created intelligence. At least that's what I'm telling them now. Realize, however, that for 25 years in academics, I was a doctrinaire, non-theistic evolutionist. I've only been a creationist for the last, 3 or 4 years. So, and I almost regret that this is being preserved on videotape. I'm not so sure I know enough about creationism to follow the party line. So if I deviate one way or the other, Earl, forgive me out of ignorance, okay? What I'm saying is I don't think we can stereotype creationists any more than we can stereotype evolutionists. There is not a theory of evolution. Evolution theory is not a monolith, okay? There are different ideas about what evolution actually is supposed to be, the mechanism or mechanisms by which evolutionary changes happen, and so on and so forth. We have our neutralists, our punctuationalists, our mutationalists, our selectionists, and so on and so forth. In the evolutionary realm, I dare say there is quite a bit of diversity among the so-called creation establishment as well. So if I don't conform entirely to Professor Hanson's predictions, that's just the way it is. Okay, this is the question we're debating or discussing, and Professor Hanson has well articulated it, so I'll just put it up here for review. And the pro statement is that evolution theory is in fact a superior scientific explanation for biological diversity. Superior to what? Superior, of course, to the creationist model. And by the way, whether or not this is scientific, let me borrow a phrase from the evolutionary literature. You know, evolutionists are very fond of using the expression, what if? You ever seen that in your textbooks? What if? What if, then, therefore, boom. By about the third or fourth paragraph, somehow it's evolved into fact. Hypothesis becomes fact through reiteration. Okay? Well, let me use a phrase, what if, when it comes to creation. What the creationist view is that whereas evolution per se brings nothing of the supernatural, theoretically, into the argument, that there's no creator, that what we have is a progress that is totally naturalistic, that is totally explicable in terms of our knowledge of chemistry, physics, and other natural laws, has brought about the sorts of biological diversity that we have on the present Earth, okay? That evolution is by common descent, and so on. Creationists said, no, it doesn't work that way. There is a theistic involvement, a supernatural creator that created. Now, that's not supposed to be scientific. Science is supposed to deal with what? Reality. The real world. What's real versus what isn't. So I'm going to borrow that evolutionist's expression. What if God is real? then that becomes a scientific form, doesn't it? If God is real, one can consider creation in a scientific format. But I haven't spent more than 5 minutes in seminary, ladies and gentlemen, so I'll leave it at that at the present time. Okay? Now, what are we really talking about in terms of the creationist model? The first diagram would represent the interpretation that came to biology a few hundred years ago in the thesis of Carl Linnaeus. Carl Linnaeus, of course, is the father of modern taxonomy. We still use Linnaean principles in our classification schemes and so on. Now, Linnaeus was a creationist, and he defined the species as immutable, unchanging. Each species was a created entity that has either remained that created entity with very little, if any, change over time, or as Professor Hansen pointed out, become extinct. So in the Linnaean view of creation, we have what my colleague, erstwhile of Harvard University, Kurt Wise, one of Stephen Jay Gould's students, by the way, now on my faculty at ICR, how about that, has termed the Linnaean lawn, where each blade or whatever represents a distinct, immutable, ne'er the twain shall meet, distinct species. This, of course, in contradistinction to the classic evolutionary model as developed principally, I guess, by Darwin and further modified over the last 100 years or so, where everything begins essentially at a single point. There may be a few fine points. If you read the microbiological literature, we start seeing polyphyletic possibilities for origin of life and that sort of thing, but we're not going to get into that this evening. In any event, starting at a more or less single point, divergence off the trunk into the various branches and so on. So evolution at this point representing change over time through descent from common ancestry. So the species is anything but immutable at that point. Everything's changing, always changing. Everything is in flux. Makes you wonder how we ever classify anything at that point. Now, somewhat off the classic creationist party line then, if you will, is what Professor Wise now refers to as the creationist orchard. Believe it or not, there are some creationists, at least two of us, who believe that things do change over time. I've got my own ontology to testify to that. Believe it or not, I look a little different 20 or 30 years ago. But that has nothing to do with evolution. Although there are days when it takes me half the morning to get up right, and some people have used that as a model for evolution, but anyway. My wife being one, I should say. Okay, the way I think, again, at least two of us creationists look at it, is in the so-called creationist orchard, where the trunks would represent what we might call baramen, the originally created kind. Now, that may not be entirely equivalent to a modern-day species. Most biologists know a species when they see one, but they're hard-pressed really to define what a species is. That's really not part of the argument tonight, so I'll just let that one wander off somewhere in the Netherlands as well. But when we say baramen, we're speaking probably of what taxonomists or systematists would regard in classification schemes as something above species level. But since various groups are classified with varying degrees of exactitude, and then we have our lumpers and splitters and so on, that variable does exist. But in the event, the trunks represent the baramen or originally developed kinds from which a microevolutionary process has obtained. So that within these major groups, there is room for variation, indeed even speciation. And much of what Professor Hansen was illustrating tonight is abundant evidence for, in fact, speciation. New species, new forms at these genetic levels of reproductive isolation, geographic isolation, hybrid exclusion, and that sort of thing. As a creationist, and I think I'm still one, I may find out differently when I get back to my room tonight, find a red light blink and say, you got a call from Henry Mars, okay? But as a creationist, I can accept the concept of speciation. What I don't accept is this major departure of ancestors into truly different forms of life, certainly into phyla, major classes, major familial groups, ordinal levels, that sort of thing, okay? So that's where our creationist concept would contrast today with the evolutionary model. Now, what distinguishes biological systems in the first place from non-biological systems is organization. And organization, of course, represents the various arrangements in which the components of a system may have. And that can be represented simply by uppercase omega, as shown in this chart. So omega represents a total number of possible arrangements of energy and matter in a given system. A given system having certain parameters that limit the number of arrangements possible through chemistry and physical laws. Lowercase omega represents the number of arrangements that actually obtain versus the total possible number. Okay? What do we see versus what could we possibly get? That relates us to the function of entropy. And the maximum entropy level, as you perhaps recall from your physics or whatever, can be related to the total possible arrangements of energy and matter within the system, modified by the insertion of Boltzmann's constant. That's what K is. Now, in a stochastic process, and what's a stochastic process? That's a process where we can't predict the outcome. The outcome is unknown from the properties of the components themselves, but we can assign a probability to any given outcome. So at that point, we can insert a probability function, P sub 0. Now, the maximum entropy level becomes a function of this probability value. See that? Anything less than maximum entropy in the system becomes a function of lowercase w. The number of permutations that actually obtain, and that has a probability P sub 1. Now, the difference between the two values, between the maximum entropy and something less, or the total number of possible combinations that are available versus the ones that actually obtain, become a function of these probability ratios. See that? It's really not that complicated. Now, for biological systems, this ratio of probabilities is related to an informational value. That's how biological systems operate. They operate on information. So where the difference in information prescribes the difference in outcome versus the total number of probabilities. And living organisms are very low entropy systems. Only a very limited number of specific combinations work versus the total number of possible combinations. There are only so many ways you can put amino acids together to give you the enzyme succinate dehydrogenase, okay? And beyond that number, certain number, 3 or 4 different kind of combinations that you get, you no longer have succinate dehydrogenase, you have another kind of protein that may or may not have a function, okay? And what prescribes that information? As we will see, it is, of course, genetic information. So in a purely physical system, we have energy and matter, which can interact to produce a maximum number of variant arrangements. In biological systems, the way in which energy and matter may interact is dictated, prescribed, and otherwise limited by information. And again, of course, that information, the archival information in a biological system is what? DNA. And where its expressed informational content be concerned, and so-called coding regions of the genes, these, of course, are represented by sequences of nucleotides. And these nucleotide sequences will prescribe certain, for example, amino acid arrangement in the ultimately translated proteins. Otherwise will be expressed in base sequences in RNA molecules, which are transcribed on the DNA templates and so on. So we have a very rigorous informational system, a system that's first ROM, or stamped into the system, or a bit of computer technology, and then subsequently transcribed through RNAs. Some of the RNAs are translated into protein structure, and so information is transduced at that point. Now, before we can really start talking about variation based on changes in genetic information as support for evolution theory, we should very briefly consider the plausibility of the genome in the first place. Now, we agreed not to get into a labored discourse on the origin of life, but this, I think, is an appropriate place to begin. If we're going to start from a single ancestral point in our evolutionary tree, we have to have some idea of the plausibility of getting that point in the first place. You agree? How plausible is it to evolve a genome from a physical environment? Well, it's very easy according to James Darnell. According to Dr. Darnell, we abiogenically brew up some particularly nucleotides in this model, and then having an appropriate pool of nucleotides, string them together. Now, our first string is not, however, DNA. DNA is simply too complicated a molecule, even for Dr. Darnell, to conjure up on a evolutionary blackboard diagram. So the consensus today, in terms of origin of the first genomes, was that the first genomes were probably RNA molecules, and there are a lot of reasons besides the complexity of RNA that favor the RNA model as a first genome. The fact that it is, at least in some instances, an autocatalytic molecule and so on and so forth, make it at the outset, plausible candidate for that. So the first genome, in any event, is hypothesized to be a polymer of ribonucleotides forming an RNA molecule. That becomes, in effect, our first gene. Then, through time, this random assemblage of nucleotides in the form of an RNA molecule becomes translationally active, begins coding for some peptide structures. Eventually, one of those peptide structures, look-a here, what if, becomes a reverse transcriptase. Now, for you students of molecular biology, that's pretty old hat. But what reverse transcriptase is in modern day biochemistry is an enzyme which does exactly that. It reverses the informational content from RNA to the synthesis of a new DNA molecule. The normal way, or the textbook way of working with nucleic acids, of course, is that we go from DNA transcribed to RNA. Here we have RNA as the archival template being translated backwards to DNA. So Darnell's hypothesis is that somewhere in the evolutionary chain of events, a reverse transcriptase evolved, which then gave us the first DNA molecule, the first genome, and then things went on from there. So from RNA through this to DNA. Sounds easy. How easy is it to synthesize an RNA molecule? Nothing to it. Just line up some triphosphate nucleotides, take out a diphosphate, and the bonds, bang, form right together. Isn't that the way it's shown in your biochemistry textbook? Yes, it is. It's shown that way on this slide. See? Here's your triphosphate. Take off the pyrophosphate here, form the bond. Right to the 3. Go from the 5' phosphate here to the 3-hydroxyl group on ribose. You've got your 3', 5' phosphodiester linkage. Do it again. You got a dinucleotide. Do it again. You got a trinucleotide, and so on. Right? That's the way it happens. Guess what? Try it in the lab. As it turns out, the second carbon of ribose, with its affiliate hydroxyl group, is more electrophilic than the third carbon. So guess what? You've got at least a 50-50 chance of forming a 2,5 bond relative to a 3,5. Did you know that? Your professors didn't tell you that. Aw, shucks. Get a tuition rebate. No, seriously. you have an equal probability of getting a 2,5 as you do a 3,5. In fact, you get a little bit more probability on the side of 2,5 because it's more electrophilic. But you knew that. Okay? So what you're going to get without an enzyme, hey, this is abiogenic, okay? Evolution. What you're going to get is in the non-biological system, in the evolving RNA genome, you're going to get at least 50% of your bonds linked 2,5 rather than 3,5. You say, so what? Here's so what. Two-5 bonds are rather stable at temperatures of 20 degrees or more below 0 centigrade. You knew that. But when you warm that thing up to about 0, guess what? The 2,5 bond shows its instability and breaks. So as soon as you warm up these molecules, which are linked at least 50% 2,5, the linkage is breaking. Guess what you ain't got any more of? No more RNA gene. Hello? Hello? They didn't tell you that in Biology 207? They will. I have faith. I have faith. Okay, so how feasible is it to spontaneously evolve an RNA molecule, an RNA genome? Here's another problem. Evolutionists tell us, and I would too, that even if you were able to put together and hold together some sort of a polynucleotide gene or complex of genes, sooner or later, to get any further evolution of the system, you're going to have to sequester it and organize it. And how do we sequester and organize such molecules? We put membranes around them, ergo cells. Now, what's the evolutionary scenario? That in the prebiotic soup we've got a lot of lipid molecules. God knows where they came from. That's not a pun. They spontaneously assemble into lipid bilayers, okay? And hey, that's a fact. We can do them in our laboratory back in New Orleans. We can actually make lipid bilayer membranes. That's okay, as long as you've got the right kind of phospholipids and so on, all right? So you can form a membrane, phospholipid bilipid membrane, and possibly, hey, what if and all that, you could do it around a held together RNA, say, gene complex, okay? Now you've got this guy organized, now he can really get down to the business of evolution, can he? You got a gene organized, sequestered away from the random environment that the cell is now in, just takes up more nucleotides, replicates itself, mutates, has natural selection act on it, voila, we're right to where Professor Hansen would like us all to be. See that? Okay? Guy starts growing hair, becomes a mammoth, what else? Okay? We got a problem. And they told you this in Biology 207 too, I'm sure, right? What about lipid permeabilities? Lipids are not notorious for being permeable to hydrophilic molecules, are they? Gang, hey, Biochemistry 101. What does not pass through a lipid bilayer with any degree of facility? Amino acids do not. Ribose sugar does not. Glucose sugars do not. Nitrogenous bases, purines, pyrimidines, and their nucleosides do not. Phosphate does not. How's this guy going to grow? How's he going to make proteins without amino acids? How's he even going to replicate his own genome without nucleoside or nucleotide precursors? Hello? This guy is off the evolutionary playing field as soon as he's got a sequestering organized membrane wrapped around him. It doesn't help evolution theory much, does it? Now, but give it enough time. Yeah, well, sure, we're talking about permeabilities that measure 10 to the 9th, 10 to the minus 11th, 10 to the minus 14th. I mean, we're not talking onrushing osmotic permeability flows here, gang, okay? But give it enough time. Evolution needs a lot of time. What happens when you sequester polynucleotides in 55 molar water over time? Biochemists, hello? Is that why you freeze dry them? Because if you don't, what happens? They hydrolyze. They fall apart. So the longer you keep that thing in that membrane, waiting for something to permeate into it to drive its biosynthesis, the longer you're given 55 molar water time to work on phosphodiester linkages and hydrolyze them back to what? Nucleotides. And you got to start all over again, right? Okay. So we have a real problem with where do genomes come from to evolve in the first place. Now wait a minute, Doc. Cell membranes are semi-permeable. Sure they are. Real cell membranes are. Why? Because they got proteins in them. They have very sophisticated protein channels in them, which may act as pores, they may act as carriers, or whatnot. But these don't spontaneously arise either. You know where the proteins come from? They're coded on those genes, right? They don't randomly just pop out of space or fall out of the primordial gumbo or whatnot. Okay? So, moving right along. Okay, what is macroevolution? What's the idea of macroevolution? Macroevolution, somehow we start with a very small gene pool. How those genes got there, we have no idea. But we start there anyway. And then through time, we expand that gene pool by mutation and selection to eventually derive the major classifications of animals and plants, phyla, classes, orders, and whatnot. That's evolution. Creation. We start with baramins, originally created kinds with large gene pools, subsequently segregate within those baraminic kinds those gene pools to produce the varieties, okay, which may be species or even higher levels taxonomically. So we start with a large gene pool and diversify. Evolution starts with a small gene pool, diversifies it through expanding the informational amount and diversifying it through mutation, and then selection acts on those variants to give us the evolutionary result. How feasible is that? Did you know natural selection was first a creationist principle? Yes, it was. Says so right here on the slide. Edward Blythe, in 1835, in fact, identified that as evidence of intelligent creator. You see how times change. I did say things changed over time, didn't I? Well, I was right. And the creator, Jehovah, Elohim, Allah, whoever, using variation within a kind to fill all different environments with all types of creatures. So natural selection is, in fact, compatible with a creationist principle. Now, important thing about natural selection, it can only select something that already exists. Natural selection is not by and of itself a creative anything. It can only act on something that's already there. Important concept. Right? Right. I don't like that one either. Dwayne Gish gave me that. Okay. What is natural selection doing reference to a variable character? Here we plot a phenotypic scale for any kind of character that can vary over a limited range. Okay, you see that? And this will be the frequency. The height of the curve will be the frequency of its expression in the population. All right, natural selection acts on populational levels of biological organization. So here we have the situation before the imposition of selective pressure. Now, applying selective pressure, the presence of an already established trait may shift the mean frequency respective of the distribution of that trait's characteristic. 3 bristles per scale to 5 bristles per scale or whatever, some variable characteristic. Switch it in either direction away from the mean. Or it may knock out the freaks, the ones at the extreme ends of the curve, such that we have a stabilizing influence here toward the previously established mean frequency. Or it may disrupt the population to produce 2 means, okay? Important thing, it's not going to vary outside these limits anyway because the traits are already established. Natural selection is only going to act on something that's there and then change the expression of that trait within the population. That is not macroevolution, however. You see that? It's important that you see that. I don't have to do that again, do I? See the dark one, see the light one, see the light one, see the dark one. I don't know where the light one went on that one, Earl. Can you find it? I can't. Okay, this is an example of natural selection acting on a previously established variable trait. Variable trait here is coloration. We've already got the genes in the population. We've got the recessive genes for the light peppered color. We've got the dominant gene for the dark peppered color. These are their mamas and daddies, dark and light, whatnot, are interbreeding and producing a population of light and dark colored moths. Then they fly around and they either land on dirty trees or clean trees or those laden with photosynthetic algae, I guess, or whatever. And then birds come along, predate the population so that the frequency of expression of that trait is going to change according to selection pressure put on it by the predator. Well, we haven't evolved light moths. We haven't evolved dark moths either. And when we change the conditions and change the nature of the selective pressure, we see a change in the population respect to the frequency of that trait. We haven't evolved vacant, as we say in Louisiana. Nothing has evolved. We've simply seen a shift around a mean value of the trait. And as Professor Hansen correctly pointed out, we still have the species of peppered moth, Bistron betularia. It hasn't evolved into a flying tarantula or anything like that. Okay, Darwin's famous finches. Darwin's famous finches are very, very similar in many characteristics, but taxonomous, being the splitters that they are when it comes to ornithology. Birds are so well known, it's very good if you can describe a new species every now and then, okay? And these birds, again, are very closely morphologically to one another, and one of the traits in which they vary by are beak structure. Hello? Oh, I'm sorry. Thank you. They vary according to beak structure, not only interspecifically, but also intraspecifically. Beak depth is particularly variable within these particular kinds of birds. And this is shown here. This is beak depth measurements for the medium ground finch species, and as you see here, the depth of the beak does in fact vary in the population, okay? Now, interesting point. Natural selection here is taking the form of relative rainfall. You have dry years and not so dry years, or wet years on the Galapagos Islands. And during the wet years, there is a decrease in beak depth. During the drier years, or periods of drought, we see an increase in beak depth in these finches, apparently because a deeper beak can crunch larger seeds, they can get more water from the larger seeds, and so on and so forth. The point is not to understand that aspect of ornithological gustatory physiology, but rather to see that we have a variable trait that is oscillating around a mean value according to subtle changes in the environment. Okay? But nothing's evolving here. That's a trait that is oscillating within the species, within the kind, according to variation in the selection pressure. You see that? So natural selection is not, again, driving a necessary basic change in the organisms which comprise the population. But we do see changes in the structure of the population itself. In fact, as Clifford Johnson has noted here, what oftentimes happens is that we see selection loss, the loss of traits through selection pressure rather than the manifestation of additional variation through natural selection. Natural selection is actually reducing variation. It's a winnowing process. It's taking a wide range of variants and constricting that variation. That's what selection is actually doing. It's minimizing variation rather than maximizing. We see this particularly, and Professor Hansen did bring this up, with respect to drug resistance and outbreeding of populations is isolated by perhaps reproductive or geographical isolation, shown in the lower figure. In the upper figure, we have a situation where we have a population with a large gene pool, which has suffered a catastrophe, perhaps a bacterial population that has been treated with an antibiotic. This segment of the population is drug or antibiotic resistance and survives. But look what happens to the rest of the gene pool. Okay, selection here has reduced the size of the gene pool. These organisms will go on and reproduce after themselves, but they're not necessarily going to increase the gene pool back to its original size. They're going to keep the genes they've got and propagate those. You see that? Same thing is true here, respect to the founder's principle, reproductive isolation and geographic isolation. Small segment of the population moves away from the larger population with the larger gene pool, carries with it a smaller percentage of the total gene pool and propagates itself. You're not expanding the gene pool, you're not changing the information, you're not expanding the information. What do you have to do to get evolution? You've got to expand the gene pool, expand the body of information, change the information qualitatively, right? Right. Glad you're paying attention. She's the one who keeps telling me, get out of the way of the projector so we can see the slide. Okay. So natural selection does not create new traits, and as a winnowing process, it actually reduces variation. Okay? So where do we get the variation? Back to square one. Start with the little guy, the point source of all of this. We've got to expand the amount of genetic information and change that information qualitatively to get the kind of variation as predicted by the evolutionary model. How do we do that? Principally through mutation. We've actually got to change the genes in order to get macro versus micro evolution. And indeed, we do see that through the so-called phylogenetic order. As we move from bacteria to, say, primate vertebrates, we do see an increase in the total amount of DNA in the genome of these organisms. Okay? Now, that doesn't mean that necessarily evolved. If this system were created, we'd see the same thing. It takes more information to run a human being than it does a bacterium. Okay? So a human would have been created with more DNA in its cellular structure than a bacterium at that point. So this doesn't necessarily prove anything about evolution, but it's consistent with the evolutionary model. If we consider prokaryotic organisms primitive to eukaryotic organisms, and we see this increase, as it were, in the amount of genetic information, that's consistent with the model. Now, one way to increase the amount of DNA in a cell, of course, is by polyploid. Double the number of chromosomes, and that does, in fact, happen. And it's happened in the case of this hybrid between some swamp grasses or wild rye or whatever they are, where they breed and initially form a sterile hybrid, but one can experimentally force them into polyploidy by blocking temporarily meiotic division with colchicine, force them into polyploidy, and then the polyploid plant is in fact fertile. Okay? And as the author correctly notes, a large number of our present-day flowering plants have derived through this aspect of polyploid. But note, as we simply multiply